Well, hello everyone. Welcome to another NAMO webinar. I'm Trent Blizzard, and I am here with Elsa Vellinga. Hi, Elsa.
Hi, Trent.
I'm excited. She's going to present to us today on parasol mushrooms and bring us all up to speed on what they are and how they work and help us do some field identifications too, I hope. I'm excited to learn about this today. Now, I'm going to turn my audio video off. I'm going to sign out and let you take over. And just a reminder to everybody, pop your questions in the Q&A and we'll get to them at the end.
Okay. Okay. Hello, everybody. Greetings from Berkeley, California. My name is Elsa Vellinga. I'm going to talk to you today about parasol mushrooms. For a start, I will say a little bit about myself. I'm a Dutch person who lives in the US, who has been living here for 25 years, 26 years, sorry. And I am a mycologist who is interested in taxonomy and what the species are in our area and how they are related to each other. Besides that, I'm a knitter. I have cats. I love to walk. I go on hiking holidays in Europe, preferably. But today I'm going to talk to you about parasol mushrooms. And there's a talk in a number of parts. We start with an introduction to the group, from what do they look like. We deeper dive into the taxonomy, which is convoluted. What do we know? Some fascinating facts, and then a short summary of everything I told. So what are these mushrooms? What do they look like? First of all, they're the most beautiful mushrooms there are. And everybody might not agree with me, but if you look at them, you know, they have everything. They have a ring, they have 3 gills, they have tissues all over the place. They can be yellow, they can be pink. They're elegant. They're just beautiful mushrooms with light colored spores. They might be green. The gill trauma, so the tissue in the gills is regular or it is all over the place from right to left, big cells coming that. In most species, there are special cells on the lamella edges that are not producing spores, but are there to protect the growing spores. When the gills are still young, that layer at the lamella edge will close the area and keep it nice and moist so the spores can be developed. The spores in most cases have a nice color change when you put them in an iodine solution. Not that they become blue, as in amyloid, but they become red. We call it dextrinoid. And then they live from dead plant material. So they're not directly connected to trees. And that means that you will find them in different forests, mostly, than an Amanita species or a Russula, for instance. So this is not a parasol mushroom. This is an Amanita. It has some characters in common with what we were just talking about, but there are differences. I don't have a photo of the whole Amanita, but you have to believe me that at the bottom there's special tissue that cover the whole mushroom, which we call a vulva. That's absent in Lepiotas, but these Amanitas have that. And these patches on the cap, they rain off. That's also a different character. That Lepiotas, what they have on top, that will stay on top. And then the trauma, the structure of the gills is totally different than in a Lepiota. You have to take my word for it. These are not parasol mushrooms. These are parasol mushrooms, again, except one, which has characters in common, I must say. The one that is not a Lepiota is up there. We go back. You look at it again. It has a lot of things in common with Lepiotas, with parasol mushrooms, but the spores are brown. So that is an agaricus species, but it is very closely related to some of those here and actually in the same general bigger group. Where can we find them? So most rich areas, the species that are most rich in species of these parasol mushrooms are forests and plantations of non-mycorrhizal trees. So in the West, in California, it is redwoods and cypresses. It is Cabeciperus further north. And then in the eastern parts, you have a lot of deciduous trees that are not in association with mushrooms. And those are also very rich, it can be very rich in Lepiotas, especially those areas where you have a little bit enrichment, for instance, along creeks, where creeks can overflow and deposit more material on the banks. Those can be really, really very good for Lepiotas, for parasol mushrooms. So let's now take a deeper dive into that convoluted history of the group as a whole. And then the first thing I show you is this complicated diagram that shows the family tree of the gilt mushrooms. And this looks a little bit different than you might be familiar with, but this is actually just a straight line, and then they bent it backwards to fit on the page. So here on the right, we have the oldest groups, and we go all the way around. And here you have the youngest kids on the block, which are the Inospis, the fiber heads. But here on the right, we have backscaps, and then we go up. The more modern species are then here, Entolomas, Amanita. And then here we have a big group up there. And that's the group I'm talking about today. That's kind of in the middle. And then the other thing that is really interesting about this diagram, this tree that shows that on the right, we have species with white spores, with exceptions of two groups, Entoloma and Plucius, which have salmon pink spores. And then here on the left, we get spores that are brown. So we have Cortinarius, Inospis, Stropharia. So brown to black spores are here on the left. But here in the middle, where the group is, where we're talking about, we get everything. We have white spores, yellow spores, green spores, blue spores, red spores, black spores, brown spores, the whole gamma. So it feels to me always like they're trying it out. And then after that, they settle just for brown. And you have to realize that a colored spore is a very advantageous thing to have because it protects the spore against drying out, against UV light. So but on the other side, it's expensive to make melanin and put it on your spore wall. So anyway, here, this is the group we're talking about. There we go. Here we are. Parasol mushrooms. And with next slide, we'll show you a little bit more about this group in a traditional diagram, just a tree on the left here. And then we see several well-known things that might anchor you in this thing, in this diagram of what we're talking about. So here, I should be able to get rid of this. Okay, oops. So here on the top, here we have Agaricus. We have Lepiota here on the bottom. We have a Coprinus here. We have puffballs here in the middle. And we have several other things, lots of names up here and up here. So what we're going to talk about today, okay, let me check this back. It's this group up here and another group down here. And the distance is a little bit, you could also, it's a little bit confusing here that these parts are so far apart from each other, because here we have all these puffballs in between, but you can also flip this part here, and then they're looking much closer to each other. But I do want to say immediately that these are not the only ones that are close to each other. So we go and look at two groups, and you see here different family names, and the jury is still not quite out whether this is the whole thing here is one family or 4 families or 5 families or 8 families as proposed here. So there's still a lot on influx, and what you can have long discussions from what is a family and things like that. So again, Coprinus comatus, so this ink-cap species is a relative of Agaricus and of Lepiotas. These Calvaceas are in the same group. Bovistas, with their small round spores, are in that same group. And also things like Tulostoma and Podaxis are in that same group. And Podaxis might look a little bit like Coprinus, but it's not a close relative. I mean, not a sister. It's more a distant cousin. But today we are here just going to talk about white spored gilt mushrooms, parasol mushrooms, spore prints, spores. That's what we're talking about. And here we have some examples of those spores. And the top we have in the Lepiotas, and another family name instead of Lepiotaceae is Vericosporaceae. So that's the top. And then at the bottom we have more of the other group, the top group we showed, though some of those types can also occur in species that are here at the bottom. But you see, these are drawn by scale. So you see small spores and you see gigantic spores. And the nice thing is that in general, small fruit bodies have small spores and huge fruit bodies have huge spores. That is one of those nice coincidences, and it's not always the case. So another picture of the spores, this time a scanning electron microscope picture. Then at the top we have chlorophyllum spores, and at the bottom we have species of spores of Lepiota and allies. So here we have these other shapes that we also just saw in the drawings. Okay, going back to that diagram with the whole group, we're going to focus first on 2 genera here on top in that upper family, the Agaricaceae in the strict sense, or the Agaricaceae in the wide sense. And I'll go a little bit longer about how taxonomy actually works. So we'll do that with the example of Chlorophyllum rhacodes up here. And that is this one. This one has a history of a lot of different names, and I don't think I've even all of them here. But it first was described at a time that everything was in Agaricus rhacodes in 1835, then went into Lepiota, then Leucocoprinus, Macrolepiota, and then finally I moved it to Chlorophyllum. And if you're a little older, then you might know that chlorophyllum, the name was only used for those species with green gills, green spores, because that's what the name means. Chlorophyllum, green leaf, green gills. So I had to defend myself and I made that change. And I'll tell you a little bit more about that. So in the past, all those species in this little tree were called Agaricus. So, and then most of them were called Lepiota, and then only one part, the one with the dark spores, was still called Agaricus. And nowadays we have all these groups separate. And then the question is, where does this belong? No, which one of the 5 names is right? And when we compare it with traditional chlorophyllum, the ones with the green spores, we see a lot of things that they have in common. They have the same kind of stem, they have the same kind of ring, they have these scales on the cap that unfortunately this photo doesn't show. The spore shape is very similar. This is the one of this one with the white spores. This is the one of the green spores. So they're very, very similar. And their DNA makeup also shows that they're very similar and actually belong in the same group. Chlorophyllum. And where they first belonged in Lepiota is down here, which is quite different from this one. And we'll see that in a few slides. So traditionally, chlorophyllum had species with green spores. So now it is enriched with species with white spores, shown here with these white ovals. And then also that species that is not opening up and doesn't look anything like those, but brownish spores also belongs there. So we see that the whole thing has changed considerably. We know more about relationships among species than we did when we only looked at the morphology we can see with a microscope or with the naked eye. So here it's really that DNA research is really very helpful to show relationships. And that's not only with mushrooms, but it is of course also with people. So all four are in chlorophyllum. So this is the one with the green spores. Here we have another picture of one of the green sport parasol mushroom. And one other thing I want to point out here is that it turns red when you crush it. The stem turns red. And that is a character that most of these in this group have. Also, this one here, you can see it down here, Chlorophyllum hortense, which is a more southern species. This photo is from Hawaii, where it occurs in grassland, in grass. But that's also one of those characters that many species have, that they turn red when the flesh, the context is exposed to air. The other big mushrooms are macrolepiodas, and those differ from their chlorophyllums by having these nice little tiger thingies on the stem. So they have separate cells there that are basically a veil. And the spores don't have this sum thing at the top, but they have a real germ pore with a little cap over it. And the spores in general are huge. And huge for a mushroom is something like 15 microns. That's absolutely huge. Very recently, at last, I should say, because we knew that there were species in the eastern part of the US and Canada that they were known under European names, but we knew they were different. But I hardly see those because I live in Berkeley. And I'm glad that people on the East Coast, in Quebec actually, and the East, have at last described these as new. So this is, oh gosh, I can't see the name, and my memory is just, this is Macellenta, I think, Macrolepia macellenta, and there's a second species, Polyda, that has been described. That is a little paler. And I think that there are more species further south than these ones that also still nameless. So keep your eyes open, collect, get the DNA done, make notes, and describe them as new. So what we just looked at were these two genera with big fruit bodies. Now we're going in the middle there. That's a group which is a little smaller fruit bodies. And here it becomes a little complicated. Some authors consider there to be two genera. Other authors say it is only one genus, Leucocoprinus. And I'll just call everything Leucocoprinus for the time being, just make it simple. Small to mid-sized fruit bodies, again, no clamps, again, metachromatic spores with turned inside blue, pink, and the outside is blue. Often a nice cuff around the stem, and the trauma is loosely organized or not. And there is huge variability in what they look like, how big they are, how massive they are, how fragile they are, what kind of colors they have. So we'll take it one by one. And the first group I want to talk about is this you can still perhaps put in Leucogericus, or you merge it into Leucocaprinus. This is a group that is easy to recognize as a group, but there are loads of species, and most of them are still nameless, I'm afraid, or there are old names that haven't been reinterpreted. So they have these long cells over the cap. They call that a cutis. So basically, it's like my hair, just long hair, long cells that are flat on the surface. That's a cutis. And these are a little bit ingrown. So this is the flagship species, Rubrotinctus, but there are lots of other ones that are similar, perhaps less orange or almost less orange. And this name Rubrotinctus has been used over the world for all kinds of different species. And I think it's only a northern, northeastern species described by Charles Peck, probably in the 1800s. So Peleus, the cap covering is a cutis, so a straight, long hair, long cells lying on the surface. Spores don't have a germ pore. The cells on the gill edges are simple. They might have crystals or not. And when you break those things or pick them or whatever, there's no color change. They stay white. And as I say, a lot of species. Here in California, there are at least 15, 16 more that need to be named. So I'll show you some of those. They're all looking pretty similar, but subtle differences. Then we go to Leucocaprinus, the rest, the main bulk of it, with, as I said, huge variation in size and stature, with or without color changes, spores with or without a germ pore. You know, it's a group where everything is possible. And this is Flammea tincta, Flammea tinctus, if this is Leucocaprinus. And this is one, when you pick it, you cannot pick it and not damage it. It turns blood red right there when you even look at it, is always what I say. And when you dry them, they become dark. And so its relatives, they have all kinds of this red reactions in various amounts. So it can be like this. It can be not in the gills. It can be in the gills. It can be more pinkish. And the nice thing about this, if you put KOH or ammonia on them, they turn green. So it's another thing that you can, a little trick you can use to identify species in this group. This is, of course, a well-known species from lawns. It's all over the place, and I always wonder whether it's native here or has been introduced with grass seeds, or who knows. Leucocaprinus leucotitis, a white, big, fleshy one, quite different than this more slender, breakable one, but still a Leucocaprinus. Spores with a germ pore, but extremely common in all kinds of, man-made grasslands, I would say, lawns and stuff. And then this is one of these fragile species that is just so amazing. This is just like a miracle in the forest when you see it. Because we don't have those in California. We don't have these in Europe. These very slender, beautiful, ethereal mushrooms that are in the eastern forests. It's just amazing species to come across. Originally described from South Carolina. A beautiful Leucocaprinus fragilissimus. So here you can see why they called this Leucocaprinus, because if you didn't know spores were white, you would call this an ink cap. So, and leukos means white, just as in leukemia, that word L-E-U-K or C. Another example of a traditional Leucocaprinus, the one that is so fluffy, Cretaceous species. A photo from Hawaii again. And then there are a lot of species that look more like this, just with brown cells on the cap. And again, here we have a lot of names, a lot of species that need names. And then also the same for these darker ones. So they are dark caps, beautiful white stems, prominent ring. And then here this character that is present in quite a few species, but is a character, these drops on the fruit bodies. It's not that these things are just out of the shower. No, these are intrinsic drops that belong to this species. And this is not Atrodisca itself. This is one of the Western other ones in that same group, but not Atrodisca. Atrodisca does not have drops, as far as I know. And unfortunately, in the old descriptions, they didn't tell you whether they were drops or not, because people made these descriptions based on often dried material, and then you don't have drops. That's a pity. And as last, this beautiful species, also from Western US, described from California, actually. And what I really like about this description, that's why I put it up, is that, oh, sorry about that. In the descriptions, it says that the type of this beautiful species was collected in Muir Woods. And you hardly see that kind of nice language in scientific literature. So it's really nice to see that here. And indeed, it is a beautiful species. Rosa olividus, widespread on the west, and a really a beauty to encounter. This brings me to another lilac pinkish species. This is Americanus. Americana. We knew that it didn't quite belong in Leucocoprinus. It is really separate, but when you analyze the genetic data, it really depends on what kind of data you use. When you use just the general most used data to distinguish species, the ITS part, it's still in Leucocoprinus, though sometimes basal, but sometimes it depends on the species you throw in there. But it doesn't fit there. It doesn't fit. It has a weird combination of characters. The spores are slightly colored, have a gempore, and they have different color reactions. They turn first yellow and then that pink, and I'll show you that later again. So this is very recently, a few months ago, put in a new genus with species like Meleagris in Macropsaliota. So it's a little different than Leucocoprinus or Leucogericus. So we have done 5 genera so far, and we move on to the next group, the lower one in Lepiotas, down here, where we have another number of species and genera. But we take this easy. We do it one by one. We'll start with the bigger ones, actually. These are, I hope you can see that in the picture, that these are pretty hefty mushrooms, something like 4 inches wide, with nice little spines on top, and a beautiful, very thin fabric of a ring here. Also here, material on the stem as part of a general veil, which is encompassed the whole fruit body when it was young. So this was split off from Lepiota and put in its own genus, Echinoderma. That was done in the pre-molecular age, and that split holds up with a caveat. But for the time, but now this is definitely a different group than the real Lepiotas, and actually it's close to sister Lepiota, these tiny mushrooms with the globose cells on it that are just totally powdery. And Sister Lepiota itself, here we have its small spores, are intermingled with melanophyllum, which is differs in the colors, but also in the colors of the spores and the colors of the gills. But they basically one group. And unfortunately, when you follow the rules of nomenclature, this would be the name for the whole group. Melanophyllum was published before Sister Lepiota. And melanophyllum is basically the same situation as with chlorophyllum. So melanophyllum means black gill, black leaf, because they turn black when they dry. And of course, Sister Lepiotas don't do that. They might discolor, they might become orange, but most of them are just white. So that's an unfortunate name for the whole group. And nobody has dared to do that yet. So, but they're intermingled, the two, and we need to make a decision. Another genus that where the species have been going into and out is Pulverolepiota. Basically the same strategy as the Sister Lepiota. A lot of loose cells on top around the fruit body, but in this case they are not globose, but they're long, like balloons, long balloons. I grew up with long balloons. Are they still? They must still be, longer cells, not round globose cells. A lot of it. And then the last group, Lepiotas. Again, small to medium-sized fruit bodies. They're actually, I would say, a bit more substantial than those in Leucocoprinus. Especially the stem looks less fragile, where you can look through. Most species have material of the veil on the stem. The spores are dextrinoid in most species, definitely not metachromatic. Clump connections are present in, again, most. And then what the cap looks, there's huge variation. There's all kinds of structures that form that layer on the top of the mushroom. And you can see here that it is really smooth because it's built up from very tightly packed club-shaped cells. So they form a layer that is just tight. And this is Lepiota castata, and its cousins, or maybe you say brothers and sisters, have a similar cap cuticle, so it's tight, so it's rather smooth, though it might be wrinkled, but it's not sticking out things. This is this beautiful Lepiota from the Cypress Grove. This is the yellow gills. Again, club-shaped cells that form this closed cap surface. Another Western species for you, another one. At last I have photos of it. And here we see something different. You see it more fluffy. So here are elements that are long and sticking out. So it's a totally different view than the other ones we just saw. And then here again, we have more of a cutis. So cells that are long and lying on the cap surface. You also see here these bands. So this is typical for Lepiotas. Most of them have these bands on the stem. This velar tissue, it's the same kind of tissue as on the cap. So it's being drawn out and it's still visible on the stem. And then another beautiful one, this that has these very fancy penguin-shaped spores. Well, narrow penguins, but a straight back, a nice belly. This is an upside-down penguin. You have to have a little fantasy to see penguins in these spores, but I hope you do it, because these are really nice ones. Again, material on the stipe, fluffy cap. And because these things have material on the stem, this is not a way to put your fingers on it. You get really yelled at by me, I'm afraid. Again, material on the stem there. So another real Lepiota, very substantial stem, quite different than what we saw in Leucogericus, leucocaprinus. Lastly, two photos of what the ones with the little spines look like. Here, this one. So these were in Echinoderma, but these have small spores and don't belong there and are in real Lepiota. So things are moving back and forth, which is a little confusing, but there must be, based on a lot of DNA data, they really don't belong with the ones we started out with here. Okay. So we've looked at a number of what we know, and the question is then, from what do we know and how do we go from here? And I want to do that with first showing from what we know and how I personally did my work to figure out from what to call things. So this map of the US shows the number of species of parasol mushrooms described per state. And you have to realize that it started in 1853 with 4 species from South Carolina. And they were described by Berkeley in England together with people in the US. So it's a different situation that changed when we look at what happened, for instance, in New York, where Peck was very active. Florida has a huge number of species described from its area. And then lastly, California. And these 3 states, when we look at the people who were instrumental in that, then it was Murrill at the first who described most species of this group. He mainly described species from the West Coast because he made a big trip, started in Washington and went down the coast. And he he lived in Florida for a long time and described many species from there. So he was actively publishing Lepiota species in this time period. And then Peck with 33 species was before him, published before him, and focused mainly on New York because he was the state botanist of New York. He also got a lot of species from other areas sent to him, which he then described. And then lastly, I myself described species from California mainly. We also immediately ask from what does the future look like? Right now, there are a lot of people who produce sequences. Sigrid is not, I forgot her name on this, people in Washington and in Indiana are very active in producing sequence data. But that's only the beginning. I want to show you how nice it is to have an old name and rediscover it or link it to a mushroom species you find yourself. I knew the name Lepiota castanescens from the literature because it's described by Murrill from Seattle. I borrowed a type specimen from New York. I looked at the type specimen. There are, these are the spores, these are the cystidia. I mean, they're really cool. And I highlighted here the colors of this mushroom. It's very unusual. It's rose, dark red on the umbo, chestnut brown when it's drying. And I could link that to this species here that is not uncommon in the West, and at least not in my area. One of the cool things about this one is that it has 2 relatives, one in Europe and one in Australia. And you wonder how evolutionary that worked. The other cool thing about this is that if you put ammonia on this, it will not turn green, but it will turn more intense red. And it also turns dark red brown on drying, as Murrill also already said. So it was for me extremely satisfying to find a name for a species you had and not to have to describe anything as new, and use a name that already exists. And I showed you on the map, there are lots of names available, but a lot of them are not used. And a totally different question I had with this one. Well, I had the same question, well, what is it? But I approached this from the other side. So I have the mushroom. It has this closed cap surface with these club-shaped cells that are tightly packed. There's nothing on the stipe, nothing. They're simple spores. And my question was really, what is it? Or does it have a name already? Or so I did first my homework, looked at the structures, so the spores, the basidia, no things no special cells on the gill edges, cap surface. This is what it looked like. I'm from the old school, so I make drawings, as you might understand. Did the ITS data, so the data that everybody used at that time. It was different from all the other ones I had, but that didn't mean that there wasn't a name available somewhere, the older literature. And for that, you really have to do detective work. So my little detective is going to look at the literature. Here we have my mushroom. We first look at the book that was available for my area at that time, 2000, early 2000s, 2005. That was David Arroyo's book, but there's nothing that fits my description, fits this mushroom in it. Then we go to scientific literature. There is an article by Sundberg about mushrooms with this Lepiota mushrooms with this kind of cap cuticle in California. Unfortunately, it's not in there, so he might have missed this species. And then I went back in time and expanded my literature search, and I came across this description by Morgan, who lived in Preston, Ohio, so not California. And he described a species that, when I read through this, it looks very, very, very similar. It has a smooth and glabrous surface. It's brown, spores are small, so I was pretty happy about that. But is it really the same is what the next question. I mean, this is Ohio, and my mushroom is in California. So I sent out looks at the type collection. I decided not to look at it in person, I think, but I looked at what other people saw on this collection. And they had published, or they made little notes about it. So the spores, what they look like, what a cap looks like. It still all looks pretty similar to the mushroom I had in mind, in hand. But then I asked Nick Money, who lives in Ohio, could you please go to this area and see whether you can find the species? Because I would love to be able to compare DNA and see it, modern collections of it. Unfortunately, he couldn't find it, but here is Morgan's photo. Here is where he lived. This is on the map. So I still have my little detective, was not quite happy yet, but he did his best. And then I got in touch with Roseanne Healy, who had collections from Iowa for something that she has also called Neofauna. And I know Iowa and Ohio are not close, but they're closer to each other than either is to California. So I looked at her collections and was able to get DNA data out of them and compare two different parts of the DNA, ITS and EF1 alpha. And you see they're close, but not 100% the same. But again, there are no morphological differences. So we are actually left with more questions whether these collections are really Neofauna or are my collections really Neofauna. So for the time being, I just call it CF Neofauna. It's not very satisfactory, but I did not have enough confidence in naming this as a new species. It's very close to what Morgan described as Neofauna. And I hope that by giving this talk today, someone will go out in Ohio in autumn and collect things that look like this and sequence them and compare the morphology and make good notes. And so we can really figure this out. There was one other loose end that still needed to be done, because in the scientific publication by Sundberg, there is the name Neofauna shows up, but this is a totally different species. It's dark, has dark scales, and this is a modern collection of it. So this fits what is called Neofauna in the other publication. So we go back. You can see from here, we can see how dark this is, and then we can look here, this dark species, which the cap surface breaks open. So this one I described as a new species using the collection that Sundberg had as the type. So we linked it back to the publication and gave it a new name. And you did it at that time, with a little Latin. You said where the type is from and what it means, and then the description. This means just that it has a dark skin. So showing these examples is a way for me to say that a combined approach is needed to tackle any group of mushrooms, but we have so many species that are nameless in this group in the US and Canada that we need to be more active in naming species. So it's a combined approach, meaning that there is literature search, macro and micro morphology have to be described, DNA work has to be done, and then to decide, is it new or is it not new? And as much as possible, also just looking at old material. And I know that it's easier for people who are at an institution to borrow original material from, for instance, Peck or Muriel, but that is in the ideal world. It's the combined approach that is needed, not just DNA, not just a photo, but everything. And yes, I am a knitter, if you didn't realize that. This is baby's first knitted DNA model. Okay, so these were the dry parts of the talk, and now we're going to look a bit at more fancy things about parasol mushrooms. And we start out with toxins. It's just to do that. There are some really, really toxic mushrooms that cause death and have caused death and did indeed cause death. So Lepiota subincarnata and its allies. So it's one group, small group within Lepiota, which has these longer cells on the cap and small, just ellipsoid spores, nice little bands on the stem. This one has these pinkish brown cap. It's pretty common, actually. And I've seen photos from basically a large part of the US. It has the same toxin as Amanita phalloides and its allies. So the death cap mushroom is the same kind of bad stuff, alpha amanitines, that cause death. So be aware that not beautiful doesn't automatically mean friendly in terms of human consumption. The other ones that are causing problems are the ones, the green sport mushrooms, green sport Lepiotas. Here's the green spore print, chlorophyllum molybditis, severe stomach upsets in gradations, but not pleasant, I have been told. I the problem is often that the green really appears very late in the development. So you might think you have a white sport mushroom, and it slowly, the green appears. It doesn't mean that the other ones are good edibles for everybody. The other white sport, I have heard people that can't eat any of those. So whether it is environmental or intrinsic to the mushrooms, I cannot answer. But this was the one you definitely shouldn't eat. Several times I mentioned color reactions, and I want to show you one more of this one where the whole fruit body becomes blue-green, greenish-blue. There are amazing changes, color changes in these mushrooms, and this is only one. There are other ways. It's not restricted to Leucocoprinus. There are also Lepiota species that turn blue, so have a blue reaction. It's really cool. That's one of the really nice things about this group. I talked already about this reddening reaction that is instant and so bright and irreversible. Look, what's it? Macrosoleota americanus starts out turning yellow first before it turns this color. So, I mean, the chemistry of this group is, I wish someone knew much more about this, but it is pretty interesting, pretty fascinating as well. So far, I've mostly talked about mushrooms with gills and a stem, but there are also truffle-like members of this group. There was one just described a few years ago, Asperosporus subterraneus. It's somewhere in between agaricus and chlorophyllum, so it's not very close, but I do want to show you that. It's grew with potted plants of a fern in nurseries in Florida. So this is what it looks like when you cut it through. And these are the spores. So ornamented spores that are not actively shut off. A really interesting way of showing it. The Chlorophyllum agaricus is, of course, another what we call sacotioid species. So this is in more dry environments. It doesn't open up. Spores, the whole fruit body withers away, and the spores are spread. And then in Lepiota itself, there are quite a number of truffle-like members, these are recent discoveries. So these three are from Utah and Colorado. So we see round spores, or these more ellipsoid spores, very thick-walled, something you don't see in the ones where the spores are actively shut off. And then this one from Spain was recently described, and it's so amazing. This color, this lilac, blue, purplish, bluish. They call it Smurfiorum, which, is little blue critters. And these are the spores. It's just amazing. And all these are in one part of Lepiota, just like this Lepiota gleba with the greenish tinges in the fruit bodies. Up here, here's gleba. So they're in this group where also this species, which I showed earlier, and Clipiolaria, for instance, belong. And up here are the other sacotioids, the truffle-like things I showed you earlier. So they all are in one part of Lepiota. And there are also sacotioid species in Macrolepiota known from Australia, for instance. So there are a few groups where you find this, but it is not all over the board, and it's very restricted. And then last, but definitely not least, the Attine ants and their cultivars. So these are ants, neotropical ant species that cultivate Lepiotas to produce their food. And again, this is one of those circle diagrams with the phylogenetic tree. The oldest ones are here. The newest ones are here. In red are the cultivars, so the mushrooms that are cultivated. In black are the free living ones. You see that these ants have cultivated quite a lot of different species. And especially these, the ones up here, the leafcutter ants, they have huge impacts on the environment. They form gigantic nests and they bring a lot of leaves to the nests and they really strip the forest. These ones, the lower ones here, they have very small nests and bring mostly dead plant material. So they have a much less influence on the environment. And so here we have a leafcutter ant in action. And this is a model of one of these nests. And they said this was a pretty young nest, was 4 to 5 years old, and it is already 4 to 6 meters in diameter. So these are all the rooms the ants had in the soil. They put clay on it, but you can imagine how big it is and how many ants there are that have impact on the forest around it. So with that, you get a little list of take-home messages. I would say yes, you can identify species to genus, but there are many species in the USA and Canada that are still nameless and or unresolved. The same for the families. But on the other hand, this is a most fascinating group in terms of spore color and color changes of the fruit bodies. There are these gastroid forms. So why did they develop in only a few genera or a few groups, a few parts of the genus Lepiota? So there's lots of questions to be answered. There are edible species, but there are also deadly toxic ones in one small part of Lepiota. And then lastly, that is the fascinating ecology for these Leucocoprinus species in relation to the Etainants. And I would say the main part from, beside that you can definitely identify species or genus, you can also contribute to the knowledge of this group in your own region. There is a lot to be done. There is a lot of literature already. It has to be linked to what you can find now. There has to be a combined DNA, morphology, and literature research, and then we can advance our knowledge of this, I would say, fascinating and beautiful group of mushrooms. So with that, I would like to thank you for being here, and thanks to all who contributed with knowledge, photos, though they might not know that I took their photos, use their photos and support in many different ways. And I'm happy to answer questions after this. And I'll stop sharing. Okay. Q&A. Okay. The first questions are about whether I prefer a larger or a smaller families. I'm still on the fence on that. At some point, so I grew up, mycologically speaking, with Lepiotas as a family, and then it became merged into Agaricacea together with Lycoperdons, and I really liked that idea. And so I am not quite sure whether I want it split up again or whether I'm happy with one large family. And the second question is about whether I looked at Lepiotoid fungi in eastern red cedar, and I did not. I did look at forests along creeks in the Smoky Mountains, but I haven't been in red cedars, and I hope that someone else will do that. The next question is from what trees do they grow in connection with? That is a difficult question to answer. So they're not mycorrhizal, so they don't depend directly on sugars from the tree. They break down plant material. And I would say they are the areas that are most species rich are those that are made up of non-ectomycorrhizal trees. So I hope I answered that question. There's one more question about the holotype of Neofauna. I did not try to sequence that because I felt very unsecure that I would be able to do so, and the material is very scant, and I didn't feel that I should do that. Question about Americanus. Yes, it should now, Leucogericus americanus should either be, I would say, is definitely not a Leucogericus nor a Leucocoprinus, and whether you call it Macropsoliota or move it into Micropsoliota, that is a question of taste. From do you want a bigger genus or smaller genera? But I would say, for the time being, call it Macropsoliota americana. And I did not know that it was edible. And you have to know your food with the correct name. So yes, it's good that you want to know what to call it. The blue reactions are intrinsic to the mushrooms. When they have a bruise, they turn blue. There is no chemical used by the human. It is what the mushroom does. And I don't know what the content is, what kind of chemical substance these mushrooms have that turned blue on oxidation.
Yeah.
Okay.
I just wanted to ask if you can make sure you repeat the questions,
Okay.
since the video won't always catch the questions. Thank you.
Okay. Sorry. Okay. The question of Joshua Birkenbach is about how do we go forward? That we are now in an age where it is very easy to produce DNA sequences, but the other skills that are needed for making descriptions and documenting species and literature research seem to be limited. And how do we teach those skills to a new generation? And what role does the amateur community have compared to the the academic realm? I think that we have to, whenever there is a foray or a meeting, that we have to sit down with people and show them how to make notes of species. That is how I grew up. I know I grew up before DNA data were available and used, but it's still a very useful skill because it also teaches you to look at mushrooms much more than just take a photo. And I don't believe in a separation between academic realm and amateur community. I've always worked together with people who are amateurs, who had very broad, very deep field knowledge of the area they worked in, or in a certain group. That there's no difference, except that as a scientist, you have access to a bit more resources, perhaps, at least literature-wise. But that's not the case anymore either, because so much is online. But I think that as a NAMA, we should pay more attention to identification skills and really looking at mushrooms and what are the characters that are important. And not just, on a foray, bring your mushrooms, dump them on the table and go and play outside. So not saying, okay, now the real mycologist can do something with it. No, it's all of us who can do that. The next question is about the ant cultivars. So ants can cultivate mushrooms? Yes. the Attine ants, so these neotropical ants, they have been cultivating mushrooms these fungi, they don't cultivate, they suppress mushroom formation. They cultivate the hyphae and they eat it. So they bring them food, and they have been doing that for millions, I think 40 million years. The first ones appeared at the Cretaceous Paleogene split. And yes, and they are the example of animal farmers, and they really manipulated their fungi. The clamp connection question. From where are the clamp connections? So clamp connections are a little thing that sticks out of one part of a hypha that connects to the next part of the hypha. And they are in general, if they're present, they're everywhere. Everywhere where you have a connection, at the base of a basidium, the base of a cell of the cap, there should be a clamp connection. And the next question is about deadly Lepiota. The common one is Lepiota subincarnata, but there are other species that are equally deadly. Spiculata is one that is a Caribbean species. There's a group of species definitely with those characters, these kind of tomatose velvety caps that break open, pinkish colors, brown colors. There's also one that pops up in flower pots with more yellow gills. It's also very toxic. So yes, okay. Oh, ooh. Okay, more a technical question about books to use when you're in the Yucatan Peninsula. From which books should you use? There's a series of modern articles by Angelini and Alfredo Justo about species from the Dominican Republic, and I might hope that these are useful for you. And otherwise it's Pegler's book about the lesser anthills, but that's further away from where you are, but there might be species in common. But there is not much known from that area. The next question is whether DNA will give the definitive classifications, or we still need to look at other macroscopic and microscopic traits in identification. That is a difficult question. DNA will tell you where something belongs, but it's a kind of a, but how easy will it be to get that? So I would say that it's also very helpful to look at the mushroom as a whole. And that DNA is only one character, and we should look at traits in general. Oh, another question about eating Chlorophyllum molybditis, and I really don't know anything about eating that. I know about one paper that was published a long time ago where 4 people were fed Chlorophyllum molybditis. One said it was delicious, one said it was okay, one said from, oh, this is very uncomfortable, and the 4th said from, oh, I thought I was dying. So individual reactions to it varied widely, and I have no idea whether cooking it in high heat would have any effect on the toxicity. And then there is an answer from, well, it's tasty, meaty, and turns your urine red. Okay, that must be about Leucogaricus americanus. Okay, this is okay. Thank you. Okay, the question is, so this, the coming fall winter of 2025 will be the focus for Russell's continental microblitz for Lepiotoids. And would this help in solving some of the problems? I hope so, but again, this needs to be done in connection with data that are available from the literature. I mean, you can sequence and you can say, oh, this is a new species, or this is a species that we didn't see yet, but it has to be linked to the morphology and to descriptions that are available. And a lot of these older descriptions, so we know that there are lots of species described from New York, but there are no sequence data for them. The ones from Florida, there are no sequence data. So if we have now a sequence, we cannot link it to a collection that was made a century ago or more than a century ago. So these are the problems that we have to answer. So we need a more, everybody has to be on board. It's not just DNA, it is the whole thing. And thank
I think
you
that
for
was the
your
last
interesting
question.
questions.
Yes,
Okay.
thank you. Yeah, a big thank you to you for spending an hour and some change with us today and teaching us a lot. There have been a lot of great comments and thank yous over in the chat. So I think people really appreciate what you brought us today.
I hope that people will focus more on these. I mean, as Joshua knows, beautiful mushrooms, and hope that, you know, and with sequence data, but it's not the only one, that the only part that's really important. I want to stress that. We have
Yeah,
still old names that have to be reinterpreted. Mm.
and I think as someone that's not an expert, in a lot of this, you really made this accessible, and
Oh,
I appreciate that. Thank you.
okay, thanks.
Okay, so I guess we're going to sign off now, and we have lots of webinars coming up over the next couple months. If you looked, we're covering a lot of different genus and species, and lots of fun. So I hope we see you all in here again soon. Say goodbye, and we'll see you soon at an event. Thank you.
Thank you.